The work you are interviewing me on is a clinical trial on CAR T-cell named KTE, which is from Kite, X19, which are CAR T-cell with a co-stimulation molecule, which is CD28, and where the binder, which is the extracellular domain of the CAR, is against a molecule which is present on leukemic cells of lymphoblastic leukemia of the B lineage, which is called CD19. So we are talking about CAR T-cell in childhood acute lymphoblastic leukemia of the B lineage, expressing CD19, which is the characteristics of all B ALL. ALL means acute lymphoblastic leukemia. So the scientists from NCI and then from Kite have derived autologous CAR T-cell, which are gene-modified T-cells, where you express this binder on the surface of the T-cell, and then you infuse this CAR T-cell to patients with uncontrolled leukemia. So we are talking about children and AYA who receive these KTX-E19 in relapsed or refractory B lineage ALL. How does it stand? The first thing is that you have to collect cells, which is the apheresis phase of the experiment. During a short period named bridge, you give generally some chemotherapy to control the disease, and then you infuse the modified T cells, the gene manufacturer modified T cells, and then this is done after a so-called lymphodepleting chemotherapy, which is fludarabine cyclophosphamide. You infuse the cells and then you wait. You wait, and generally happens some reactions which are called cytokine release syndrome and sometimes neurotoxicity, and I will come back on that. And then at some point, the disease at day 28 should be controlled, and the topic of the study was how to define the best dose with this product to be used in refractory B lineage ALL. So in fact, during this trial, which is the phase 1 of a more ambitious trial, which is currently in phase 2, during the phase 1 of this trial, 3 doses of cells have been tested. The first one was 2 million per kilo of CAR T-cell expressing cells. The second one was 1 million, but this 1 million was in a volume called 68 ml. And the last one was 1 million in a shorter volume, which is 40 ml. And at the total, 24 patients have been treated. Median age were 14. 4 children have been treated at the 2 million level, 11 at the 1 million 68 ml level, and 9 children at the 1 million level. These children have been all pre-treated very heavily for their previous leukemia, including having received transplant in 1/4 of the case, including having received already immunotherapy targeting CD19 in 1/3 of the cases. And one child, if I remember, one child has received also another immunotherapy, which is an antibody drug conjugate called inotuzumab. All of these children at screening were in blastic phase in the marrow at 44% in median. And all these children were essentially blastic immediately prior to the lymphodepletion, 37% in median, with a range between 0 and 100%. So the question when you infuse CAR T-cell is, what is the toxicity, what is the efficacy? Toxicity was relevant, and the main toxicity that we are anxious to see is so-called cytokine release syndrome. It was severe, which is grade 3, 4, or 5 in 1/3 of the patients. Fortunately, no deaths have been registered, which is grade 5 in this trial. And interestingly, no grade 4 have been registered. Grade 4 means threatened to life, life-threatening, sorry. And none have been seen in this trial. Only grade 3 have been seen, which means that a lot of children have had fever, more than 70% of the children, and hypotension, which is 2/3 of the patients. Generally, these effects happen very quickly, which means that the median time to onset was 5 days. And generally, these effects last for 1 week, which was the case, by the way, in this study. During the CRS, cytokine release syndrome, can happen also the fact that the cells can penetrate the central nervous system and give some clinical adverse event. And we already know that with this CD28 CAR, this is a frequent event. And in total, 21% of children, which is 5 children, have experienced grade 3 or 4 neurotoxicity during this trial. In terms of symptoms, those neurotoxicity can correspond to confusional state, or encephalopathy, which is approximately half and half for all these kids. In terms of efficacy, the response in terms of overall complete remission, or so-called CRI, which is complete remission with incomplete recovery of the counts, have happened in 67% of the cases, which is 16 here. And no response was seen only in 8%, which is 2 kids. Interestingly, also, the depth of these response were very important, since even at the so-called MRD level, which means that you detect in a very sensitive way the presence or absence of leukemic residual cells, in 75% of the cases, which is 18 children, we have not seen any more disease at day 28, generally was the assessment. What have been seen also in this trial have been that there was a big expansion, what we know about CAR T-cell already, followed by a drop, which drop means that essentially you cannot detect any more CAR T-cell after approximately 8 weeks. So after 2 months after infusion, you don't see any more CAR T-cell. There is a certain trend between the expansion, the degree of expansion, or the number of copies of CAR T-cell that you detect, and the toxicity, the CRS. But essentially, the neurologic event was here. There was a trend because the p-value was 0.06 for severe neurotoxic events if you have a great expansion of the CAR T-cell. There was also a trend toward a best response in terms of MRD. The more you have CAR T-cells circulating and expanding, the less disease residual you have after those CAR T-cell administration. to summarize the findings, we could say that one, these CAR T cells have induced high rates of MRD negative remissions and high rates of response in children and adolescents with refractory relapsed B-lineage ALL. That with a median follow-up, which began to be long because it was 3 years in all treated patients, the median overall survival was not reached, which is a very important in such advanced phase children in terms of severity of disease. All the AEs, the adverse events, were consistent with what we know about CAR T-cell toxicity. And finally, which was interesting for the rest of the study, obviously, is that it was defined as a compromise between efficacy and toxicity, that the 1 million per kilo dose concentrated in a small volume, which is 48 ml, was the best dose selected for the phase 2, which is currently ongoing in France and in, not in France only, sorry, in USA and in Europe, including France, by the way. And interestingly also, we have adapted the phase 2 for including even first relapse when they are very early, which means after less than 18 months after the first diagnosis. So in the phase 2 trial, we will have the same population that was in this phase 1, plus a little less advanced kids, but with a very severe disease because their relapse occurred as a first relapse within 18 months from diagnosis. And there will be also, during this phase 2, other disease, which is lymphomas, which are rare disease in terms of relapse in children, because most of the kids with relapse refractory NHL of the B-lineage are cured at the first attempt by chemotherapy. So we will have rare children with lymphoma, DLBCL or Burkitt, and or primary mediastinal large B lymphoma in this trial. Thank you very much for your attention.